Module 7, the nervous system. Action potentials 2, stimulus and response. It's time to finally talk about how a signal gets sent down the axon of a neuron. First, an action potential is an all or nothing occurrence. This means that there aren't stronger action potentials or weaker ones. Neurons either fire or they don't. Second, an action potential cannot be produced until it has a stimulus. That means that excitable cells like muscle cells or neurons can produce action potentials. When you learned how muscle cells contract, you learned that a neuron produces a neurotransmitter called acetylcholine and sends it to the postsynaptic membrane of the muscle cell. That causes an action potential in the muscle cell. Well, the acetylcholine is the stimulus for this action in skeletal muscle cells. But there are other neurotransmitters that can stimulate action potentials in cells. Neurotransmitters are released at a synapse. And just like there are synapses between neurons and muscle cells, there are synapses between nerve cells too. So neurons can pass action potentials from one to another by sending neurotransmitters across those synapses. Sometimes an environmental change can cause a stimulus. When you pet a soft kitten, your fingers are pushing against its fur. You can feel how soft the fur is because the movement of the receptor cells in your fingers pushes up against the fur and sends action potentials to your brain so you could identify the sensation of soft furriness. Light can also create a stimulus in certain cells in your eyes, causing action potentials to travel along the optic nerve to your brain. How about when you pick up a mug of hot cocoa that's too hot? Heat receptors in your fingers cause action potentials to travel up neurons to your brain so that you know that you need to put that mug down. Chemicals can also create stimulus in cells of your tongue so you can taste food. Well, an action potential is basically an interruption of the resting potential. At resting potential, there's a difference of negative 85 millivolts. It's negative because as compared to outside the cell, the inside is negative. When a stimulus occurs, such as you picking up that hot mug of cocoa, the potential difference changes. As you can see on this graph of an action potential, the potential difference changes from negative 85 millivolts to positive 30 millivolts in the axon. What caused this? Well, as a result of a stimulus, whether it's light or heat or touch or chemical, depending on the specific cell, channel proteins within the plasma membrane open up. These channel proteins are specific for sodium ions, so they can travel across the membrane. Now, there are already a lot more sodium ions outside the membrane than inside, so if the channel proteins are open, the sodium ions will freely travel from outside the cell to inside the cell. And that makes the inside of the cell more positive, causing the potential difference to change. In a fraction of a second, so many positive sodium ions move into the cell that the inside of the cell is now more positive than the outside of the cell. So the potential difference is now positive. The sodium ions are rapidly drawn into the cell for two reasons. First, the concentration of sodium ions outside the cell is greater than inside the cell. And second, the inside of the cell is negatively charged due to the greater positive charge on the outside, and also the largely negatively charged proteins inside the cell. The sodium ions can freely diffuse into the cell because those channel proteins opened and allowed them to. But if sodium ions diffuse in, they'll stop moving into the cell when there's an equal number of them on the outside as the inside. Yet there are still many more potassium ions inside the cell than outside. So once the sodium ions have evenly distributed themselves outside and inside, there's still a greater positive charge on the inside now because of the greater supply of potassium ions there. This process is called depolarization. Now, polarization means there's a difference in charges between two areas, and there indeed was a positive charge outside and a negative charge inside. Well, depolarization means there's no more separation of charges. It technically means there'd be no net charge and the potential difference between the two areas would be zero or neutral. But because so many sodium ions moved into the cell, the potential difference rises above zero, up to a positive 30 millivolts. The process is still called depolarization though because positive charges were finally allowed to move inside. But the action potential is not complete. The axon has to get back to its negative resting potential, and this signal has to happen rapidly so the system can run efficiently. If it took a long time to send a signal, the system would not be so efficient. Imagine if it took a very long time for the signal from your fingers to reach your brain, telling you the mug of hot cocoa was burning your skin. You could get burned. So to get back to the resting potential, the sodium ion channels close and different channel proteins open up. These channel proteins let potassium ions move through the plasma membrane. There are still way more potassium ions inside the cell than outside it. So as the potassium channels open, potassium ions quickly move out of the cell via diffusion. This makes the inside of the cell less positive because it's losing more and more positive ions. The potential difference then gets lower and lower until it becomes negative. So many potassium ions move out of the cell that the potential goes back to negative 85 millivolts, and even a little more negative than that. This process is called repolarization because the charges are separating again. Well, though the charges are back to the resting potential state, the ions are all mixed up. Sodium ions are inside the axon and potassium ions are outside. This had to be done to repolarize the cell quickly. That's why the sodium ions didn't just move back outside again. But we now need to get everything back in its original place. This is done via the sodium-potassium exchange pump. It pumps sodium ions outside the cell and pumps potassium ions in. Summing up, at resting state, the sodium-potassium exchange pump has used active transport to move sodium ions outside the cell and potassium ions inside. It moves more sodium ions out than potassium ions in, so there's a net negative charge inside the cell and a positive charge outside. Also, the cell has produced large negatively charged proteins inside. All this creates a resting potential difference of negative 85 millivolts. The sodium ions cannot move inside the cell, and very few potassium ions can move out. A stimulus causes the sodium ion channel proteins to open. Sodium ions freely move into the cell because of the greater concentration of them on the outside, plus the pull of the negative charges inside the cell. This results in a potential difference of positive 30 millivolts. This part of the action potential is called depolarization. Next, the potassium ion channel proteins open, causing potassium ions to move out of the cell. They do this because there's a greater concentration of potassium ions inside the cell, so they want to diffuse out. Plus, the inside of the axon is positively charged, so the potassium ions are repelled. The potential difference is restored to a little more negative than negative 85 millivolts. This part of the action potential is called repolarization. Finally, after repolarization, all the channel proteins are closed, and the sodium-potassium exchange pump begins to do its job, moving potassium ions into the cell and sodium ions out. And all of this happens in a few milliseconds. It's truly a fabulous design.